Hi, I'm Soufyan Lakbir, a PhD student in the group of Sanne Abel and Rebel Feynman, and today I will present our work on CIBRA and computational method to identify genomic alterations with a system-wide impact. Cancer is characterized by many genomic alterations, from single nucleotide variants to copy number aberrations and structural variants. However, only a few alterations drive tumor progression, and from the many alterations present in cancer, we don't yet know their impact on tumor biology. Traditionally, frequency-based methods are used to detect potentially biologically relevant alterations. These methods assume that the onset of genomic alterations in the genome are more or less random, and that an enrichment is an indication of selection/biological impact. However, there are confounding factors that can influence the mutation rate across the genome, leading to false positives. And additionally, these methods are mostly only focused on single nucleotide variants and cannot detect the impact of structural variants. In our work, we propose to utilize an alternative data modality to assess the impact of genomic alterations. We hypothesize that biologically relevant alterations induce a characteristic change in the system that could be measured from the transcriptome. So, if a genomic alteration is relevant, it will induce a specific change in the transcriptome that we can measure from the transcriptomics data to identify their impact. To assess the system-wide response of genomic alterations, we first perform a differential expression analysis between our genomic alteration of interest, the cases, and the control group. We use expression data to perform this differential expression analysis, and we fit and bet a uniform mixture model to the p-value distribution generated from the DE analysis. We then take the integral between the beta and uniform component to assess the extent of change in the system, and we then also perform a permutation test to assess the significance of this change. To assess the capability of CIBRA to identify known biology, we performed a pan-cancer genome-wide screen on coding single nucleotide variants in primary cancer. I show on the right the results of the screen, and then mostly only of the top 20 hits, where on the x-axis we have the cancer types and on the y-axis the different genes, annotated for if they're already a known driver. In color is the impact score indicated, the CIBRA impact score, and the size indicates the number of cases. What we can observe is that CIBRA is able to identify the majority of tumor suppressor genes and oncogenes as having a significant system-wide impact. We also assessed if CIBRA was able to identify novel biology by performing a genome-wide screen on coding single nucleotide variants, somatic copy number aberrations, and structural variants in metastasized colorectal, breast, and lung cancer. And what we surprisingly observed was that many genes affected by structural variants showed a significant system-wide impact, as can be seen here, and even to a similar extent as known drivers as p53 and RB1. This shows that that structural variants actually have a large biological impact that has thus far been largely underreported. And moreover, it also shows that CIBRA is able to detect novel biology. While we can assess the impact on a gene level, as what we have done before in the genome-wide screens, it is also important to consider the type of genomic alteration. Therefore, we examined whether we could dissect and identify the relevant subcategories of mutations based on both their type and location. When we refined the definition based on coding type for 4 example genes, APC and p53, which are known tumor suppressor genes, BRAF, which is a known oncogene, and Titin, which is a gene often mutated in colorectal cancer, and this data is actually for metastatic colorectal cancer, we observed that when we do this refinement for BRAF, we see that missense variants are the only variants relevant with a significant system-wide impact, and the non-coding variants do not actually induce any system-wide effect. And when we take the combination of these effects, so if we didn't do the refinement, we observe a drop in signal. So this shows that the refinement actually is important to actually detect BRAF as a significant driver gene. But we can also see that even if we do this refinement for a gene as Titin, there's actually no improvement in signal. And this shows that if there is actually no effect, the refinement doesn't matter. For APC and P53, we actually need to do an additional refinement. As they're both tumor suppressor genes, we need to add an additional layer, and this layer is copy number loss. And when we include this additional layer of copy number loss, we observe that the combination of coding point mutations and copy number loss results in the highest impact. And this you can, for example, see for P53 as the combination of nonsense of frameshift variants and the copy number loss results in the highest score. We can also observe this for APC, as also a nonsense of frameshift and a loss induced the highest impact score. In summary, CIBRA is able to identify cancer drivers by combining genomic alterations and bulk RNA-seq data, and we can actually do that with as few as 10 cases and controls. We also unexpectedly found that many genes affected by structural variants have a significant systemic impact, and this shows that the impacts of structural variants has been grossly underestimated. Moreover, we have also shown that CIBRA has the potential to refine current definitions of genomic alterations in terms of their impact. I would like to acknowledge all the groups involved, and I'm happy to take any questions.